Hi, I'm Blair Sheehan, and in this series of slides, we'll be talking about blood cells and blood plasma. So the cells are erythrocytes or leukocytes. About 99% of them are erythrocytes or red blood cells, and approximately 1% are the leukocytes or the white blood cells. Leuko for white. The plasma is made up as that part of the blood that's not the cells. So it contains mostly water, ions, glucose, gases, oxygen, carbon dioxide, amino acids, etc. Basically, it's all of the small stuff in the blood. It's all small, and it can leak out of the capillaries through the walls into the ISF, which we'll talk about more later. The albumin proteins, part of the plasma, are important for osmosis. So if you remember that albumins are osmotic proteins, that would be good. We'll talk about those later when we talk about capillary exchange, where fluid leaves the capillary and then is drawn back into the capillary. Clotting proteins are pretty self-explanatory. They're involved for clotting blood, keep you from bleeding excessively. And the immunoglobulins are other proteins in the blood, and these are the antibodies. They're globular proteins of the immune system. So the albumins and the clotting factors are made in the liver. And the immunoglobulins are made in white blood cells. WBCs. And plasma cells and B cells that we'll talk about more when we get to the immune system. Move this out of the way. So this slide is about plasma, and if you need to prepare a sample of plasma, this is basically how you do it. You start with a blank tube with nothing in it on the left, but you have to add an anticoagulant. And that will prevent the blood from coagulating or clotting. You don't want the blood to clot if you're trying to prepare plasma. You fill the tube with blood, so it has blood and the anticoagulant, and then you centrifuge it. And you spin it, and that causes the heavy material, the cells, to settle to the bottom. And what you wind up with are several different fractions of the blood. And at the bottom are the red blood cells. So there are red blood cells, and then the top is the plasma. So you've separated the red blood cells, pulled them out, and in between the two, the red blood cells on the bottom, the plasma on the top, is this very thin layer that's referred to sometimes as the buffy coat. Buffy because it's sort of yellow cream colored, and that's where the white blood cells are and the platelets. So you'll see that as a thin sort of white cream colored film between the plasma and the red blood cells at the bottom. And this essentially is the hematocrit that we'll talk about shortly. So that's how you make plasma. Put whole blood into a tube containing the anticoagulant. That has to be in there to keep the blood from clotting. Okay, and then you just spin it, separate it out, and you get those three phases. For the hematocrit, which is just that percentage of blood, red blood cells at the bottom. Oops. This is showing three different hematocrits. So they took blood from three samples and added an anticoagulant to each one, whole blood, spun it, and then separated it out. So this tube on the left shows a normal hematocrit, and in in females, it's about 37 to 47% of the volume is the red blood cells. In males, it's slightly higher than in females. This is due primarily to higher levels of testosterone in male blood. So again, the plasma, the buffy coat, and the hematocrit. If there are insufficient red blood cells, so here that there's a depressed hematocrit, the hematocrit is lower than it should be, that's a type of anemia. You don't have as many red blood cells to carry the oxygen in the blood. And there are many types of anemia. On the other hand, if you have an elevated hematocrit, there's more than normal amount of red blood cells, and that is an example of polycythemia. Generally, it denotes a problem in the bone marrow where the blood cells are being produced. So three examples of hematocrit: normal, anemia, and an example of polycythemia. On the other hand, if you don't want plasma but you want serum, you prepare it a little differently. So it's important that you know the distinction and the difference and be able to define the difference between plasma and serum. So you know how to make plasma. Here's how to make serum. You start with a tube again, and in this case, you do not put in an anticoagulant. So there's no anticoagulant. So the whole blood is collected and it is allowed to clot. You just leave it out on the bench at room temperature for about half an hour, and then you simply spin it. And after you centrifuge it, the red blood cells go to the bottom, as before. Red blood cells plus the clot. And then you're left on the top with the upper phase, similar to plasma, but the difference between plasma and serum is that in serum there are no clotting proteins. They have been removed in the clot because you allowed the blood to clot before you spun it. So that's the main difference between serum and plasma. Next slide reinforces that. So unclotted whole blood on the left, and there are the red blood cells and the plasma and the buffy coat. On the right is the clot and the serum on top. So what pulled out, what fell out when you spun it, what separated out was the clot, which is made up of the red blood cells, of course. The platelets get tangled up in it, and so they pull out. The white blood cells also tangled up in the clot. They're separated out, and the clotting proteins. That's what formed the clot. Clotting proteins are sometimes referred to as clotting factors. Basically the same thing. So, what's the difference? What are you going to say about serum? How could you define serum? Well, you could say that it's basically the plasma minus the clotting proteins. There are other proteins that are still in the serum, such as the immunoglobulins or the antibodies. And if you're following this COVID-19 outbreak, which I imagine you are, one of the discussions is in making a blood test to find out who is immune to COVID-19. So after the patient has been infected, if they recover, they will have antibodies in their blood which are in the serum. So they refer to this testing as serological testing. And they're looking for the presence of antibodies. So I'm not going to talk any more about serum. Right now we'll do a little bit more with antibodies when we get to the immune system. But just sort of FYI, when they're talking about anything serological, they're referring to the serum of the blood. The last slide is for you to practice calculating an hematocrit. It's pretty straightforward. This is an example or a lab that we do. If we were meeting in person, you would be given a capillary tube, which is a small, thin glass tube, and you basically fill it with blood by touching it to a drop of blood on a microscope slide. And as the capillary tube touches the blood moves up the capillary tube by capillary action, and you can fill the glass tube. And then you would put a plug in it because it's open at both ends, and you would put a clay plug so that it doesn't come out, the blood won't come out, and then you spin it. And when you spin it, the capillary tube has an anticoagulant in it. Since we're going to collect plasma, you don't want the blood to clot. And when you separate it, you have the three phases. There's the plasma, and then the packed red blood cells. And the hematocrit is equal to the percent of packed red blood cells. In whole blood. That's our hematocrit. So you spin it, and then you just figure out the percentage. So you would take a ruler. One way of doing this is to just take a ruler, and you're going to measure the height of your column of liquid. This was the same height as your column of blood. And then you're going to also measure the height of the red blood cells. And if you call the height of the red blood cells, you can measure this in millimeters. If you let that be A, I don't know, let's say 26 millimeters. And then you measure this distance. We call that B. Well, it's just a straight percentage, a straight proportion. So the hematocrit would equal A over B times 100, because you want it in a percent. And that's about all there is to it. So calculating hematocrit, you ought to practice that. Make up a little drawing, change some of the numbers, and figure out, well, you're going to have to figure out what B is equal to. Just add them up, and that would be a 100% of your volume. Hope that makes sense. Ask questions if it doesn't.